The first thing to note is that cervical dystonia is part of adult onset focal dystonias, of which there are a variety of forms. When it involves the neck primarily, it would be called spasmodic torticollis or cervical dystonia, but focal dystonia can also involve the eyelids, which is called blepharospasm, the vocal cords, spasmodic dysphonia, or hand and arm, in which there would be writer's cramp, musician's cramp, or occupational cramps of various kinds. All these focal dystonias are in fact related to each other from a point of view of genetics and pathophysiology. Now, in terms of the clinical aspects of these dystonias, the main features are abnormal movements, twisting movements, pattern movements, sustained postures, and in relation to spasmodic torticollis, of course, that involves the neck. The muscle groups that are involved, the agonists and antagonists, that is, the muscles that pull in one direction and pull in the other direction, are involved simultaneously with these movements. It's patterned in that the same types of postures occur over and over again. The speed can be slow or rapid. Sometimes it can be very fast and very jerky. Quick movements are called myoclonus, and it can be even in that speed. Types of actions usually worsen the abnormal postures. But of course, in the case of cervical dystonia, one's always using the neck to support the head, so it's always active in that situation. It may vary with body position. It can be rhythmical as well as jerky, as we talked about before. And as almost all neurological conditions, it can be worsened by stress or fatigue, and it can be improved by hypnosis, relaxation, sleep, sensory tricks, and we'll talk about sensory tricks a little bit more in a moment. So you all know what cervical dystonia looks like, of course. Here are a couple of photos, and I'll show you a couple of videotapes as well. This one is from YouTube. You can find anything on YouTube these days. And I'll show you this one.
I'm going to make like this, and every time I try to bring it up, I just right back down again. And I try to hold it still, and just so far straight. I feel like I should.
So here you see the head tilting over to the side, a little bit of rotation as well, and difficulty in maintaining it in an upright position. And here's another example.
Jerky torticollis, cervical dystonia.
And just let you can do it, it's going to be. Okay, now look up at me. Try to straighten your head out.
This woman's head is twisted, with the chin turned to the right shoulder, tilted with the left ear pulled to the left shoulder, and with jerky movements, particularly as she tries to overcome the pulling of the muscles.
Okay, no, that's good enough.
You see the abnormal posture and also the jerky nature or rhythmical jerking that she has in the head. So there are a whole variety of forms. And just one more videotape.
Cervical dystonia. This man has a tilt of his head with turning of his chin to the right. Cervical dystonia is a focal dystonia of the neck musculature and is commonly known as torticollis.
Pointed out when I was not aware of it. Sometimes I do this also.
He demonstrates a sensory trick in that when he raises his arms or touches his chin, there is a reduction of the torticollis.
Yeah, I don't have the feeling.
It stops the flow. Okay. Now you can put your arms down.
So that deals with the issue of sensory trick and gray matter in the thalamus, the caudate, superior temporal lobe, and the left cerebellum. That's where these dots are. The caudate is part of the basal ganglia, and the thalamus is a relay station of information from the basal ganglia to the cortex. So this involves certainly a number of structures that are related to basal ganglia, but also the cerebellum, another part of the brain where there appears to be an abnormality. These are increases and then decreases in the putamen. Putamen is also part of the basal ganglia structures. So there are increases in some parts, decreases in some parts of the basal ganglia, but also involvement of cerebellum and other parts of the cortex. So there are a variety of areas here that are of potential interest. Another study that was published recently, which I think is an interesting one in relation to trying to identify abnormalities, and this was published in 2009 by a group that are based in Ireland. And they looked at, again, voxel-based morphometry. And what they did was to try to identify a marker for relatives of patients with cervical dystonia that might carry the same genetic background, but that haven't come down with the illness yet. And they use a marker of sensory abnormalities. Now, it turns out that there is a mild sensory abnormality in patients that have cervical dystonia. It's very mild and something that's not ordinarily appreciated, but quantitative testing will in fact reveal it. And it turns out that relatives of patients some relatives of patients with cervical dystonia also have the sensory abnormality. So it's possible that the sensory abnormality is something that indicates a basic genetic abnormality, but only some people will get cervical dystonia, okay? Because it's a combination of things that we have to get. So the sensory abnormality may be an indicator of a basic underlying gene defect. And what they did in this study was to look at the voxel-based morphometry to see if there was something that one could find that was special about the patients that everyone's asymptomatic, don't have cervical dystonia, but some have sensory abnormalities and some don't. And basically you can see from the title here that what I found is that basal ganglia abnormalities were seen in the unaffected relatives of patients, in some of the unaffected relatives of patients with cervical dystonia. So this is what they did is they looked at a large number of families in which there were patients that had cervical dystonia, some of which had other types of focal dystonia as well. And they looked at the ones that did not have cervical dystonia in those families. This is looking at the sensory score. This is the unrelated control subjects that all do well here. And then there are the unaffected sporadic relatives and the unaffected familial relatives. You see some are in the normal range and some show the same abnormality that's seen in patients with cervical dystonia. So if they then compare the brain with voxel-based morphometry in these patients who are abnormal to these patients which are normal on the sensory test, they find abnormalities, again, largely in the basal ganglia. So there are voxel-based morphometry abnormalities in basal ganglia regions similar to the types of voxel-based morphometry abnormalities that I was showing you in the patients with cervical dystonia. So this suggests that there might be some genetic influence that leads to an abnormality of some sort in the basal ganglia, perhaps, that is a possible substrate or one of the factors that will lead to cervical dystonia in the long run. This is looking at the amount of voxel-based morphometry change and the change in sensory score. The more that there was an abnormality of sensory score, the more abnormal was the basal ganglia abnormality. This now goes back to the Obermann study that I showed you before with the cervical dystonia. I'll have a chance to answer your questions later, okay? I'm close to the end. This is looking at patients with blepharospasm as opposed to cervical dystonia. This was in the same Obermann study that I mentioned before. And blepharospasm patients had abnormalities in similar places that the cervical dystonia patients did, both in basal ganglia and in cerebellum. Those regions had some changes. But exactly what those changes represent, yet we still don't really know completely. All right, the final topic I want to cover is pathophysiology. What do we understand about the way that the brain is malfunctioning in order to cause these abnormal postures? And in terms of the physiological studies that have been done, there are three prominent types of abnormalities. Deficient inhibition in the brain. Basically, the brain is organized to have excitation and inhibition, increases and decreases, which are normally balanced. And if there's a loss of inhibition, then things tend to be too excited, and you get too much. And basically, there's too much muscle activity in patients that have cervical dystonia. And it turns out to be deficient inhibition rather than excessive excitation that leads to this problem. There is disordered sensory organization. I've already been talking about the fact that there are these mild sensory abnormalities that can be identified, and this is due to abnormalities of the way that the sensory system is operating as well as the motor system. And then there appears to be increase in brain plasticity. Brain plasticity is the important function of the brain that allows changes because of environment. People learn to do things because the brain is plastic. If you learn something that you didn't know before, then the brain has changed. The brain has developed that particular capability. But there are some ways in which changes in the brain are not helpful, and they can go in the wrong direction. And there are certain aspects of increased changes in the brain in patients who have different types of focal dystonia, and this can be one of the problems as well. Say a little bit more about each of those. There are many ways of looking and proving that there's a loss of inhibition. There are a variety of reflex abnormalities. There are cortical abnormalities that have been shown a lot with a technique called transcranial magnetic stimulation, which looks at the excitability of the cortex. There is a loss of a neurotransmitter, inhibitory neurotransmitter in the brain called GABA, GABA-aminobutyric acid, which has been shown with a different magnetic resonance imaging technique called magnetic resonance spectroscopy. There's a loss of dopamine D2 receptors that has been shown with an imaging technique called positron emission tomography, and this probably leads to a loss of inhibition. And we know therapeutically the drugs that increase inhibition can be therapeutic, like the benzodiazepines. And as I mentioned before, you will hear more about therapy later. In terms of sensory organization, there does appear to be some disorganization of the somatotopy in the primary sensory cortex. The sensory cortex of the brain is ordinarily laid out in a very standard pattern. One can look and see that the representation of the thumb is here, the index finger there, the middle finger, fourth finger, ring finger, all laid out very nicely. Somehow that gets mixed up in patients that have focal dystonias. It doesn't come as nicely, neatly laid out. And this gives rise to abnormalities that one can see in sensory discrimination. As I mentioned, they're very mild, but they're pretty definite. Everybody finds them who actually looks for them. There has been, I didn't show you, as I said, voxel-based morphometry studies. There are many of them that show different things. There is at least one voxel, there are several voxel-based morphometry studies that show abnormalities in the sensory cortex. I was showing you one that showed primarily basal ganglia and cerebellum, but there are other voxel-based morphometry studies that show sensory cortex. And there's one study that shows increasing sensory cortex activation with increasing amounts of dystonia, suggesting that the sensory cortex is very much involved. And then there's increased brain plasticity, which is the third point that I mentioned. And this has been demonstrated with a relatively complicated technique using transcranial magnetic stimulation. This is a method called paired dissociative stimulation, or PAS. And PAS is a certain type of transcranial magnetic stimulation technique that was meant to mimic a basic science method for looking at the nature of synaptic plasticity. Changes in plasticity occur at multiple places, but one of the important places is at the synapse, the sort of junction between different neurons. And one of the types of changes at the synapse is something called long-term potentiation. And PAS is meant to be a technique that you can apply in humans to look at the cortex, to look at a method, to look at something like long-term potentiation. And this is increased in individuals that have focal dystonias. So there is some abnormality of plasticity then that can be seen in this situation. Okay, so the bottom line then is that focal dystonias arise due to hyperexcitability of muscle contraction leading to the abnormal postures that appears to result from the combination of some sort of permissive substrate like a loss of inhibition, like an abnormality in the basal ganglia or cerebellum that we saw in the voxel-based morphometry, and an environmental factor such as trauma or other things that might trigger it, giving rise to some sort of aberrant plastic change in the central nervous system control mechanisms for movement. So it's a combination of multiple factors that will give rise to the situation. It's not going to be simple. One hopes that the situations will be simple, but it turns out that it's going to be multiple complex factors that give rise to cervical dystonia in the end.